Thank you very much, Mitchell. Our final speaker in this panel is Gary Gold, who is a professor of radiology and biomedical imaging, professor of bioengineering and orthopedic surgery, chair of the Department of Radiology at Stanford University.
Good morning. So I'm going to start out by mentioning that my only disclosure is I get funding from GE Healthcare. But my other important disclosure is I'm perhaps the only faculty member in my department that does not have a startup. And most of those startups are in AI or diagnostic technology. So this slide is borrowed from Mitch, actually, a while ago, just emphasizing the importance of integrated diagnostics and the fact that radiology and pathology are well aligned to achieve this. And I think prostate cancer, in this case, work from Mirabella Russo, is a perfect example of the marriage of advanced imaging technology with MR and pathology to achieve better diagnoses and better outcomes for patients. Now, I was fortunate to work with Sam Gambier for over 10 years, and Sam was a friend and mentor, and he introduced me to the term integrated diagnostics. We were trying to come up with a new name for the Department of Radiology, and I was at a swim meet with my son, and Sam was on the phone, and I said, Sam, it sounds like you want to merge with the Department of Pathology. And in true Silicon Valley fashion, Sam said, no, I want to acquire pathology. But Sam did some really great work early on with integrated diagnostics at Stanford, looking at non-small cell lung cancer. Two studies here. The first one looking at circulating tumor cells, which were weakly correlated with PET markers of disease. But then the second one looking at circulating tumor microemboli, which were much better and actually improved the diagnostic accuracy compared with either of the markers by themselves. Sam started this baseline study, which is now run by Ken Mahaffey, which is really a study of 10,000 normals with all of the tests we have available to basically answer the question, what is normal? With all of these tests. This includes genomic, multi-omic analysis, imaging, and clinical reference points. And this has been a common theme today of multiple silos. And part of my talk today is a bit of a mea culpa, coming in as chair of the department after Sam. I realized that I've been missing opportunities to push the field of integrated diagnostics forward. And it's a lot because of silos. Here's a good example. This is some work from our nuclear medicine molecular imaging division looking at PSMA, gallium for prostate cancer. Concomitantly within our own department at the Canary Center for Early Cancer Detection that Sam founded, we have another one of our scientists, Tanya Stoyanova, developing a novel in vitro biomarker for prostate cancer. And these studies were done at the same time. And afterwards, when I discovered this, I'm like, did you guys ever think about talking to each other and working together and integrating these things? And of course, the answer was no, because at Stanford, we have a saying, let 1,000 flowers bloom, which basically means that every PI is independent. So we have novel, fast imaging, MRI that can be done in 1 minute. We have theranostic agents from our nuclear medicine division, in this case for neuroendocrine tumors. And we have new in vitro diagnostics, which are really enabled by the Canary Center, which Sam founded, which is, I think, pretty unique in a department of radiology to have a group of scientists and physicians developing new in vitro diagnostic tests. Here's an example of one of these tests using a chip to detect extracellular vesicles for early cancer detection. This is another one, and this one, as a former engineer, this makes me crazy that I didn't think of this first. This is basically to increase your yield of circulating tumor biomarkers. You insert what amounts to a sticky wire, and you capture the circulating tumor cells through multiple passes of the circulatory system, increasing your sensitivity. This came out of Sam's lab. This is another one out of Sam's lab. This is one of our residents using another novel integrated diagnostics approach, volatile organic compounds. So you breathe in DNA mini circles, they get incorporated into the lung cancer. They have a promoter gene on them to produce limonene, and the limonene is breathed out and detected. And that's being correlated with PET-CT here. So I think of integrated diagnostics very broadly and includes the things that Sam thought of with wearable and in-home technology, which will be further parts of this ecosystem that we've been talking about today, where pathology and radiology data will be integrated. We're going to have home-based monitoring and wearable monitoring. And this is a good example. This is the smart toilet developed by Sam's lab that would basically monitor your urine and stool in your house for early signs of disease. That data has to be integrated with the other data for actionable decision making. We have more advanced techniques being developed by Joe DeSimone and Sharon Pateri. This is a novel way to collect and analyze interstitial fluid on the fly with a patient who wears a patch to look for evidence of circulating tumors or proteins in the interstitial fluid. And this has sponsored a company by two of my faculty called Eigen, which they have these wearable sensors with microneedle technology that you wear as a patch on your arm, and molecular recognition of particular molecules using aptamer technology that'll continuously look for signs of protein in the interstitial fluid. Outside of cancer, we've got examples of integrated diagnostics going on in the area of head injury. This is for concussion, looking at combination of imaging and pathology. And then in my field, we have high-resolution fast MRI and optical biomechanics with IMUs and motion capture to look at the relationship between gait and the development of osteoarthritis. So this is going to come in every field, not just cancer. So take-home points, cancer, as Sam always used to say, is detected too late when it's most difficult to treat. And this integrated diagnostics approach really gives us the promise to detect and treat cancer early and properly. And in-home and wearable technology gives you more opportunities for detection. So I do want to say the silos that everyone showed this morning really resonates with me, because As chair, I have silos within my own department, research groups that should be talking to each other to take advantage of opportunities that weren't talking to each other. So I consider it my job to fix that. Thank you.